Hey y'all, Miss Mayhew here, and today we're going to talk about nucleic acids. In this video, we're going to be focusing on Chapter 3, Key Concept 3.1. This is where we're going to talk about the function of nucleic acids and the fact that they are these informational macromolecules. So, reviewing some of the basics of nucleic acids, we're going to start in the first row here, and you are going to highlight or circle the elements that make up nucleic acids. The elements that you should have highlighted are carbon, hydrogen, oxygen, nitrogen, and phosphorus. Next, you're going to go to the second row and you are going to highlight or circle an example of nucleic acids. The box that you should have highlighted or circled is the DNA, RNA, and ATP box. The third row, you are going to underline or circle or highlight the type of bond that is between nucleotides. The box that you should have highlighted or circled is the phosphodiester bond. The next row, you're going to highlight or circle the purpose of a nucleic acid. And the box that you should have highlighted is the box for genetic information. The last row, you are going to highlight the monomer of nucleic acids. And the box that you should have highlighted are the nucleotides. Let's talk about some examples of how the structure affects functions in nucleic acids. Okay, the first example I'm going to use is I'm going to talk to you guys about the double helix structure of DNA. Now, DNA, typically you're used to seeing it look something like this. Now, what that represents are the nucleotides in two strands facing each other and held together by hydrogen bonds. So if you zoom in, it looks a little something more like this. Now, those hydrogen bonds are important because they're relatively weak, meaning that this DNA strand can be opened up and unzipped for a lot of processes that we're going to talk about later in the year, including protein synthesis and DNA replication. A second example that I'd like to share with you guys is the shape of the nitrogenous bases within each DNA molecule that are going to allow the DNA molecule to remain straight. That's because the base pairs that align with each other are consisted of a purine and a pyrimidine, which we'll talk about later in the video. Basically, though, this allows the spacing of the molecule to keep those phosphate sugar backbones straight. You have a single ring paired with a double ring every single time. If you have a double ring and a double ring, that would cause the DNA to bow out. If you had a single ring and a single ring, then that would cause the DNA to bow in. So because these base pairs are aligned with each other and form these hydrogen bonds in a specific way because of their shape, then that allows the DNA molecule to be packed properly. Now, nucleic acids are the polymers. The monomers of nucleic acids are nucleotides. So a bunch of nucleotides are able to link together to form a nucleic acid. And there are two different types that we talk about: DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. These nucleotides are made up of three things: a pentose sugar, meaning a 5-carbon sugar, a nitrogen-containing base, and a phosphate group. A nucleoside is a pentose sugar and a nitrogen-containing base without a phosphate group. Now, these nitrogenous bases can be categorized in two ways. We have pyrimidines, which have single rings, and purines, which have double rings. One of the things that differentiates DNA from RNA is the type of 5-carbon sugar. In DNA, it's deoxyribose, and in RNA, it's ribose. And just like the name of these sugars implies, the difference between them is the lack of or the presence of a oxygen. In this diagram, you can see the 3 components of the nucleotide. These orange boxes represent the nitrogenous bases. The pentagons, the blue pentagons, represent the 5-carbon sugar, and the yellow circle represents the phosphate. On this table, we have a comparison chart between purines and pyrimidines. Remember, these are the nitrogenous bases. Purines, like I said, are double-ringed structures. Pyrimidines, on the other hand, are single-ring structures. They would look something like this. Purines are consisted of adenine and guanine, while pyrimidines are cytosine, thymine, and uracil. Now, these nucleotides are joined together through a condensation reaction, again, the formation of water. We also refer to this as dehydration synthesis, meaning to take water out to form something. Okay, these nucleic acids, as they're joined together, can only grow in the 5' to 3' direction. And that is all because of the location of the hydroxyl group on the 5-carbon sugar. When you line up 2 nitrogenous bases on top of each other, like so, the 2 hydrogens and the oxygen that we're going to take away to form our water droplet and our phosphodiester bond between these 2 nucleotides is located here and here. So now we're going to remove 2 hydrogens and an oxygen, forming a water molecule, which will leave us with 2 nucleotides linked together by the remaining oxygen. So let's go over some comparisons between RNA and DNA. The first one is that RNA is single-stranded while DNA is double-stranded. The second one is within the bases that they contain. RNA contains adenine, uracil, guanine, and cytosine, while DNA contains adenine, thymine, guanine, and cytosine. The next one is in their 5-carbon sugars. RNA has the ribonucleic acid, meaning it has that extra oxygen on it, and DNA contains the deoxyribose sugar. Now, like we've mentioned in one of the examples at the beginning, these base pairs are paired up complementary, meaning they fit together just right. Okay, these base pairs you need to know. They are between adenine and thymine, or in the case of RNA matching, that thymine would be placed with uracil, and cytosine and guanine. They always pair together. You also need to know how many hydrogen bonds are between each complementary pair. Those adenine and thymine pairs only have 2 hydrogen bonds, while the cytosine and guanine have 3 bonds. A little kitschy way to remember which ones are paired together, I always think of Austin, Texas, great city. A and T go together, G and C go together. So on this slide, I'm going to go ahead and label the parts of the DNA molecule for you, just as kind of a review. Okay, the first thing I'm going to point out is the phosphate sugar backbone. Notice that they run anti-parallel to each other. And here we have the complementary base pairs. And also would like to point out that this molecule is a double helix, meaning you have 2 strands winding around each other. That's it for this video. Remember, if you have any questions, make sure to ask your teacher.